Welcome to Inside Pathology. Today, we begin at the very foundation. What is pathology? Pathology comes from Greek. Pathos means suffering. Logos means study. So literally, pathology is the study of suffering. In medicine, it means the study of disease. A long time ago, Virchow said, "All disease begins in cells." And he was right. When cells are healthy, the body is healthy. When cells are injured, the body suffers. Cells live in balance. We call it homeostasis. But when stress comes, cells must respond. Sometimes they adapt. Sometimes they get injured. So the cell is the foundation of health and disease. But cells don't act alone. They are guided by molecules, genes, proteins, metabolites. Where do these instructions come from? From the genome. The human genome, sequenced at the start of the 21st century, a landmark in science. Today, sequencing is cheap. Computers are powerful. Omics tools, genomics, proteomics, metabolomics are everywhere. They are changing how we understand health and disease. The genome is not just a linear code. It is a complex system of regulation. The human genome has 3.2 billion base pairs. Only 20,000 genes make proteins. That's just 1.5%. Surprising fact, a worm also has about 20,000 protein-coding genes. So, what makes us human? Not the number of genes, but how genes are used and regulated. 98.5% of the genome does not code for proteins. It was once called junk DNA, but it's not junk. It regulates gene expression. It is the blueprint. It is the architecture of life. Humans are 99.5% identical in DNA. The differences are in just 0.5%. These differences make us unique. They influence how we respond to disease and environment. Two common variations: SNPS, single nucleotide changes; CNVS, copy number variations. Some are good, some are neutral, some affect disease risk. Together, they explain diversity in humans. DNA sequence alone cannot explain everything. Even identical twins look different. The answer lies in epigenetics. What is epigenetics? Epigenetics means changes in gene expression, without changes in DNA sequence. It's control above genetics. DNA is 1.8 meters long, yet it fits inside a tiny nucleus. How? By wrapping around histones, like thread on a spool. This creates chromatin. Heterochromatin equals dense, silent. Euchromatin equals open, active. Only unwound regions can be transcribed. Histones are not static. They carry chemical marks. Methylation effect depends on the residue. At some sites, it recruits transcription activators. At others, it recruits transcription repressors. DNA is negatively charged because of phosphate backbone. Histone tails are positively charged because of lysine residues. Opposite charges pull them together. DNA wraps tightly. Acetylation neutralizes lysine charge, reduces attraction, DNA loosens. Acetyl groups are also recognized by bromodomain proteins, which recruit transcriptional activators and co-activator complexes. Deacetylation restores positive charge, DNA tightens, transcription silenced. Phosphorylation can open or close chromatin, depending on the site and context. DNA methylation typically silences genes by blocking transcription at promoters. These marks are reversible. Writers add them, erasers remove them, and readers interpret them. Clinically, histone deacetylases inhibitors and DNA methylation inhibitors are being developed to reactivate tumor suppressor genes such as P16 that are abnormally silenced in cancer cells. Not all RNAs make proteins. Some RNAs regulate genes. Two main types: microRNAs, long non-coding RNAs, MicroRNAs are short, about 22 bases. They silence mRNA after transcription. They control development and cancer pathways. One microRNA can regulate many genes. Full cRNAs are longer than 200 bases. They can silence genes or enhance transcription. Example, XIST. It coats one X chromosome in females, wrapping around it like a magical cloak, making the entire chromosome fall silent, almost as if it becomes invisible. CRISPR was first discovered in bacteria as a defense system. When a virus called a phage infects the bacterium, it injects its DNA. The bacterium saves small pieces of this viral DNA in special regions of its own genome called CRISPR loci. These act like a memory bank. Later, if the same virus attacks again, the bacterium makes a guide RNA from that memory. The guide RNA joins with Cas proteins, such as Cas9. This complex patrols the cytoplasm, finds the matching viral DNA, and Cas9 cuts it. In this way, the bacterium has acquired immunity against phages and plasmids. Scientists borrowed this bacterial system and use it as a powerful tool in human cells. They deliver Cas9 together with a guide RNA into human cells, using carriers such as viral vectors or nanoparticles. Inside the cell, the guide RNA finds the exact DNA sequence we want to target. Cas9 then cuts the DNA at that location. The cell now tries to repair the break. Sometimes it just sticks the ends back together, often making small mistakes. This can switch a gene off. Other times, if we provide a correct DNA template, the cell uses it to repair the break accurately. This allows us to fix mutations, replace faulty genes, or even add new ones. In short, CRISPR is a molecular scalpel, precise, programmable, and revolutionary. Cellular housekeeping. Why housekeeping? Cells stay alive by doing everyday chores. Protection, nutrition, metabolism, communication, movement, energy, cleanup and renewal. We call this cellular housekeeping, compartmentalization. Many chores are separated inside membranes. Why? To keep harsh enzymes contained. To create special environments. Low pH, high calcium. This makes reactions safer and faster, making proteins. Proteins for secretion, membranes, or lysosomes start in the rough ER. They're folded and checked. Then the Golgi edits sugars, stamps them, and ships them to the right addresses. What about proteins that stay inside the cell? They're made on free ribosomes. Smooth endoplasmic reticulum. Builds steroids and lipoproteins. Detoxifies hydrophobic drugs. Stores and releases calcium. Cells must recycle. Three main sites do the breakdown: proteasome, lysosome, peroxisome. Proteasomes. Proteasomes digest tagged proteins. Ubiquitin is the tag. Short peptides go out. Some become antigens for immune display. Lysosomes. Lysosomes are acid recyclers. They digest macromolecules and old organelles. Autophagy brings worn parts in. Phagosomes bring microbes in. Peroxisomes oxidize very long-chain fats. They generate hydrogen peroxide. Then catalase breaks it down into water and oxygen. Endosomes sort incoming cargo. Secretory vesicles deliver to the surface. Cytoskeleton moves everything and keeps cell polarity intact. Mitochondria are best known as the powerhouses of the cell. They make ATP through oxidative phosphorylation, using electrons from nutrients to drive proton pumps and harnessing that gradient with ATP synthase. But mitochondria do more than energy. They supply building blocks for biosynthesis, like precursors for amino acids and lipids. They are also the site of heme group synthesis, critical for hemoglobin and many enzymes. And mitochondria are guardians of cell fate. They sense stress and damage, release cytochrome C, and trigger programmed cell death, apoptosis. So mitochondria are not just batteries. They are decision makers, balancing life, growth, and death. Organelles are not permanent structures. Each has a limited lifespan. For example, mitochondria last only about 10 days. Before a cell divides, all organelles must be duplicated, so each daughter cell inherits a full set. But old or damaged organelles cannot remain. They are recognized, targeted, and removed by processes like autophagy. This constant renewal ensures that cells stay healthy, efficient, and ready to adapt to changing conditions. The plasma membrane is not a rigid wall. It is a dynamic fluid bilayer made of phospholipids. Each phospholipid has two parts: hydrophilic heads that face outward into water, and hydrophobic tails that cluster inward, away from water. This arrangement creates a selective barrier, keeping large or charged molecules from passing freely. But the membrane is not uniform. Different lipids are enriched on different sides, outer versus inner leaflet. This asymmetry is crucial for cell signaling, recognition, and interactions. Special membrane lipids. Phosphatidylainositol. Phosphatidylainositol is more than a building block. On the inner leaflet of the plasma membrane, it acts as a scaffold for signaling proteins. When phosphorylated, it generates second messengers, like DAG and IP3, that relay signals deep inside the cell. Phosphatidylserine. Normally, phosphatidylserine stays hidden inside, giving the inner membrane a negative charge that helps attract certain proteins. But when flipped to the outer surface, it becomes a dramatic signal: eat me. Macrophages recognize it, marking the cell for clearance during apoptosis. Glycolipids. On the outer leaflet, glycolipids dominate. Their sugar chains extend into the extracellular space, helping cells stick, communicate, and interact. They play roles in immune recognition, sperm-egg fusion, and inflammation. In essence, they are the cell's social antennae. Membranes are barriers, but life needs exchange. Small molecules slip through. Ions and nutrients use channels and carriers. Some cargo needs energy, active transport. Cells connect through junctions. Tight junctions seal the space between them. No leaks allowed. Desmosomes anchor cells together, giving tissues strength and resilience. And gap junctions. They communicate. Tiny channels that pass signals and ions from one cell to another. Together, these connections form unity. This is how tissues hold, and how life stays connected. All this harmony, membranes, organelles, skeleton, connections. This is what keeps life in balance. This is the cell, the smallest unit of order in pathology. If you enjoyed this video, don't forget to like and subscribe for more. In the next episode, we'll see what happens when this harmony is disturbed, when the cell faces stress, injury, death, and adaptation.